From this, we have our RNA comes and RNA polymerase, which are different proteins that comes into the nucleus and unwind the double helix that the DNA has, and start reading specific sections of the DNA that later will be translated into proteins. So one specific change that we have from DNA into RNA is that our nucleotide timing is changed with uracil. As you can see here in this example, we can see how here we compare the A up here with the A down here, but then instead of having a T, which corresponds to thymine in the DNA, we're having RNA, the nucleotide called uracil. And this is one of the major changes from DNA to RNA. At the chemistry level, we can see that from thymine to uracil is pretty much exactly the same, but we just are missing our carbon group. This is essential because it will allow us for the RNase polymerase to come and to be able to translate into proteins. How DNA actually is composed is made out of pieces or chunks of letters that we call exons and introns, and how these exons, they contain the essential knowledge to have the proteins. And introns in our DNA, they are work in sense to stabilize or to make more stable our DNA structures. When we are translating or transcribing from DNA into RNA, what happened is that, as I was telling you that in the analogy of the recipe, that we try to compact or better store those precious recipes, that's exactly what our cells are doing from DNA to RNA. They come and they splice these intronic regions that then no longer have a function because at this point we have RNA and we don't need the stability that DNA requires. And after removing these intronic pieces, all these exons are joined together to create something that we call the messenger RNA. From the messenger RNA, we've been talking so far that everything is happening inside of the nucleus. Transcription from DNA to messenger RNA, it happens in the nucleus. And from this point on, this messenger RNA is taken out to the cytoplasm to be able to be translated. This messenger RNA, they are composed into, they have the coding information to call for essential amino acids, which in our body, we have 20 of them. And how they go is that every 3 letters of the messenger RNA that we have is called a codon. And those 3 letters that we have there, we can pair them as our first letter, our second letter, and our third letter here to follow this table to be able to decode for every single amino acids that we are calling in our DNA and RNA. And now, like, to make it a lot easier in academia, we know every single of those amino acids, and we have them here as a 3-letter code or 1-letter code, and this is used in various scenarios where people prefer to have them as a 3-letter code or 1-letter code. So pretty much since we have 20 essential amino acids, pretty much we can create any type of message coming out of the DNA and code them into those specific amino acids. Based on that, I have created a specific activity for all of you who are here present with me or they're in Vegas or doing online. And I kind of want to just take a few minutes to see if we can decode this message that I have hidden here and see if we can go all the way from DNA to messenger RNA to the amino acid sequence and see if we can read actually what is happening. Just to help you here, remember that from DNA to messenger RNA, every T that we have is changed with a U, and then everything else stays the same. So maybe we take 1 or 2 minutes so that you can actually see if you can do that, and I'm just going to disclose how the messenger RNA looks like. Here on campus, I can see some people already finishing with their messenger RNA. So I hope that you get something like this, where every T was exchanged with a U, and everything else remains the same. So from here, as if you remember what I said, how we go from RNA to the amino acid sequence of that peptide, every 3 letters, it comes together to forming essential amino acids. And if you come and you get UGG, that will give you from the table that you have in your paper, it will code for a W, just to give you this, like, and then you go on and on and on. And how we read that table, once again, is we get our first letter here from our left side, and then our second letter from here on the top. So if I will go and get U, and then if I need, my third letter is a G, in that case, then I just come right here to do U, G. And that's how we read that letter. And then we get here that amino acid would be leucine, and leucine in this case, for this specific example, it's an L, and then you can write L whenever you need it. So we can take a few more minutes and then I can disclose what the message actually is saying.
Our guests here in Nevada are working hard.
Yeah, sorry for putting you onto the test. Okay, I think most of you are wrapping up with your hidden message here. So we can just take one more minute and then. Okay, how are we doing now? Should we disclose what it says? So with all that amino acid sequence that you have, you should be able to say, welcome to Rossman, if you follow the instructions. So hopefully it's close enough with what you have right now. But yeah, this is the process that is happening in our cells every single second that we have, that we live. So from all these essential amino acids that we come, and then just this as a game, right? How do we create proteins? After that, RNA comes out, a messenger RNA comes out of our nucleus to our cytoplasm. We have these specific molecules, they are called tRNA or transverse RNA. Those transverse RNA, they are bound to those specific amino acids or those essential amino acids that we've just been talking. And these tRNA, they complement to our messenger RNA. And when they complement to each other, so for example, I told at the beginning that A always pairs with T, or in this case, A will pair with U because we are talking about RNA right now. And then when we're having AAA, and we have a tRNA called UUU, then that's a lysine amino acid. And same for on and on and on. So in our cytoplasm, all these tRNAs are trying to come and form all those peptides. They come and they grow this peptide change of amino acids. And this peptide change are what form our proteins essentially. Sorry. So our proteins are, what is our protein structure? Well, all these essential amino acids, they come and they form this peptide sequence. And then they come, and based on their chemical structure, based on the hydrophilicity or the hydrophobic interactions that they have, meaning if they like liquid or they don't like liquid, as we are made out of mainly water, they come and they form these structures that they call this alpha helix, kind of like a ribbon, or these beta sheets, kind of like arrows going up and down. And they're starting to twist to each other to create tertiary structures as that peptides continue growing until they just come and they form like a big protein, which it will have a specific and essential function. And this is how we go all the way from DNA to proteins. And in all this process, we call it central dogma. And then for all of you who are familiar with a little bit biology, you know that we just covered a whole semester of how this actually works. So good job, you passed. Now, let's talk a little bit about how DNA reveals our heritage. Now that we understand how DNA is actually goes has the essential concept and the essential information of life that creates us, that creates our cells, that creates our organs. How is it that they actually comes and reveal our heritage? As I said, we're a society where we bond with family. We are people who have some social interactions. We tend to have a family, and then we procreate, divide, and then we have our children. But our children, as you can see, there as people who have kids, our kids, they never look alike, like it's identical to us. They always look a little bit like grandma, a little bit like grandpa, a little bit like mom, a little bit like dad, sometimes a little bit like uncle or aunt. So why is that? How is it that it's working? How is it that if mom and dad, why we don't have a hybrid of like half looks like mom, like half looks like dad? We will talk a little bit about that. Also, something that I wanted to talk is the DNA traces our human history in migration times. If you have ever had the chance to take one of those DNA tests, 23andMe or any of the other ones, they will come and tell you like you have 20% of, in my case, like European, 80% of Native American, because they don't know how the whole Americans are divided. And like 0.2% African. And in my head, I go like, African, how is it that Africa comes into my blood? It's just because we believe, or we know by now in science, that human society, they evolved in the east portions of Africa about 200,000 years ago, and they started to migrate from there into Europe, Asia, Australia, and Oceania, and then eventually they move into the Americas, going down to South America. That information is stored in our DNA, and we can trace it back. For all of you who has, I don't know, like European descendants, then they can even trace even better, like, okay, you have this percentage of your Irish or German or and all of that, because we have traces that are specifically, they works as a signature of those specific places where we come from. And we know this because in an event that always happens in our DNA or in our cells, that we call it DNA recombination. As I said, 50% of our DNA come from each of our parents. In this specific example, parent A, biological parent A, in this case, it will be dad and mom. They have, as I mentioned at the beginning, we have 2 copies of the chromosome, right? Like solid and a little bit like shaded here on pink, and with mom, blue solid and shaded blue in this one. So when we pass our DNA into our children's, our DNA, we don't come like, are we just going to take this whole block of DNA and put it into child 1, and this whole block of DNA and put it into child 1 as well to create mom and dad, that comes from mom and dad. The DNA likes to recombine or switch places from places. So there is always a little bit of a mix of mom and dad with grandparents, just as it's saying here. So in this specific example, we had from mom, we have 1 recombination event, meaning half of DNA jumped from one side to the other one to create this chromosome here. And from mom's side, we ended up having 1, 2, 3, 4 recombination events where different pieces come and they create each other. That's the reason why we tend to look like a little bit like mom, dad, grandpa, and grandma, because we always been creating all those recombination events. So this recombination is a biological process where segments of DNAs are broken apart and then rejoined, resulting in new combinations of genetic materials, essentially creating genetic diversity. That is essential. And that is essential because history taught us that when we don't have those recombinations and when we try to become as pure as possible, we tend to have problems. A classic example is hemophilia. As some of you might be familiar with this type of disease. This was nicknamed as the royal disease because in the 19th and 20th centuries, when England, Germany, and Russia and Spain, they had their royal houses, they wanted to maintain their royal blood as close as possible. And what happened is that Queen Victoria, now we know by tracing back, she had this rare genetic disease called hemophilia, which is an inheriting bleeding disorder that prevents blood from clotting normally, meaning you bleed out. You don't form clots in you. And then because they were pretty much inbreeding within families, they were passing this disease over and over and over generations to the point that pretty much everybody here, as you can see, some of the, they're highlighted kind of like in orangey. All of the relatives, they ended up having this disease, and most of the women's in the royal family, they died after birth because they bled out, and that was terrible. And we also see this type of events of when we have that much inbreeding also in animals. Another example is cheetahs, for example. Cheetahs, they tend to be protected, and when they have an autoimmune disease disorder, and pretty much any virus that comes into cheetah animals, they tend to die. Pandas is another example. Have you seen, if you had the chance to see how the people that take care of pandas in YouTube, or if you're going to the zoo, they suit up completely, like they're going to go into space, because they're protecting those panda bears from getting any viruses or any vectors that we might be given to them. Why? Because they have an autoimmune disorder. They will not be able to survive if they get a disease, as well as little fever. So this is really important to try to understand like how the DNA actually comes and works and take place in all of this. And why is that important? Well, in our little example here, what happened if we just come and we decided to change in our fourth codon this C to an A? And then not a big deal, right? It's only one letter out of the 3 billion letters that we have in our DNA. But what happened when we come into our messenger RNA? Then all of a sudden instead of coding for the whole message saying welcome to Rosman, then we would just hear and we'll say welp, and then our cells will say like, wait a second, you're telling me to stop. I will not continue to produce the protein or the message that you want me to produce, and I'm going to be short, and then that protein might just be lost. And because of that loss of the protein, then diseases might be able to happen, or risk for something else might be able to happen. To these nucleotide changes, we call them SNPs, or single nucleotide polymorphisms. And these come in 3 different flavors. They come either as a missense mutation, which is when the SNP changes the codon code to a different amino acid, just like the example that we just saw. A nonsense mutation is when the SNP creates a stop codon and leading to a truncated protein, or a silence mutation, when the SNP changes the codon, but it still codes for the same amino acids. So as you can see, a nonsense mutation is a lot more harmful than a silent mutation and a missense mutations, obviously. And you can tell me, Dr. Gonzalez or Josue, how is this really a big deal from all the proteins that we're producing by minute in our cells? How much is a big deal of this? And I just wanted to have this a little bit experience. All of you could just complete it in your piece of paper, the first activity. Now I want you to, like, for the multiple times that you have done this, I want you to create a airplane, a paper plane, following these instructions, and see how. And then we'll put into test what is the best airplane, how they can fly. So take a minute and let's just do some origami to understand science. And I'm sure that some of you don't even need instructions, right? You have kids, and every day as soon as they remember that you can make uh, paper planes, they just ask you like, Dad, Mom, make me one. And your house is just full of paper planes all over. At least my house is. So for this example, you have the essential material. You have your messenger RNA, which is your paper. You know exactly the instructions that are, like, the step by step of how to make the paper plane. Now we need to put it into a test. We're saying that one specific amino acid can change the outcome um, of how the paper plane will fly. Um, so I would just ask for 1 or 2 volunteers to, at the same time as I count 1, 2, 3, and you can do it as well in Nevada, in the Nevada campus. Just as soon as you're done with your paper plane, just throw it and see how it flies, and see if they fly the same. I see a few of them going. Some of them they fly really well. Some other ones they don't. Let's go. Like some other.
Who wants to fly?
What changed? We have the same paper. I mean, at least here on campus, we have the same paper. Everybody followed the same instruction. What's different? What I like? Yeah, some of you are saying like the person who did it, the how you actually fold it, the plane, all of that. Sorry, how we throw it. Yeah, exactly. So even though we have the same instructions, even though we have the same materials, every single environment that we have, that we call it in this case, like how we throw it, how we fold it, how like did we follow the instructions to the dots, or did we create any mutations because we said like, maybe I don't going to follow this instruction, I'm just going to fold it my own way that I've been doing it for the past, I don't know, 10 years. All of that changes. That's exactly what's happening in our proteins as well. Every mutation or every DNA change that we have changed those proteins ended up folding. And in some cases, it can be, it might be functional, just like all the planes here, they flew, but it might not be efficient, meaning not all the planes flew the same way. Just like in our proteins, not all the proteins might be able to function to the perfection. And that brings us to the next step, which is DNA and health. And a classic example in biology is sickle cell anemia. Sickle cell anemia is caused because a single point mutation in the gene called HBB. And that single point mutation over here, as you can see here, it changes from an A to a T. And that changes the amino acid that goes from glutamine to valine. a single point mutation, a single fold that we did a little bit too much or a little bit too less, and then we develop this disease. And what this disease causes is that it changes the shape of our red blood cells, and they cannot longer function properly. They cannot carry oxygen as efficient anymore, and our red blood cells become rigid because it changes the structure of our cells, and it because it creates this valine. It's a more hydrophobic amino acid, meaning it doesn't like to be surrounded by water, and then it just hides and compresses that protein a little bit more, making it more rigid and not being able to actually function properly. And this is just an example of one single mutation to cause this disease. This mutation, it's needs to be present in both copies of our DNA from mom and dad. If we have only one bad copy, our body can still function normally because it just make up from the bad copy with the good one. If we have two bad copies, then we ended up having this mutation. However, when life gives you lemons, right? This mutation is really frequent it's found frequently in the equatorial length of the planets. So Central America all the way to a little bit like north of South America, Africa, Asia. That's where these mutations, or people has this mutation present the most frequently. Why? Because in those parts of the world, we have those mosquitoes they transmit malaria. What is the malaria? Malaria is a parasite that comes and gets host on our red blood cells to the point that eliminates them. So why having a bad copy of this gene, or a mutated copy of the gene, helps them against malaria? Well, the host no longer can go into the red blood cells, so the malaria germ doesn't go and it dies. So for them, that's something useful to have. So they pass that copy of DNA that for us will be bad. They pass it into their children so they can be protected because they live there. So this is a good example just because, it's bad for regions of the world where mosquitoes is not a problem. Utah, for example, we have winter that kills everything. But in the equatorial lane, where we have a forever green area of tropical and beautiness, then where we have a lot of more insects and stuff like that. Another example that I like to present, especially here in Utah, is the breast cancer, especially with BRCA1 and BRCA2. If you're familiar, a couple years ago, Mira Genetics, which is a company which is really close to the U, they were doing a lot of DNA testing. They were trying to understand how breast cancer works. And they were trying to understand, like, okay, with all these mutations coming up, is there a mutation that in these breast cancer genes that produces cancer? And they come up with a series of tests, and they come up with a series of mutations that, yeah, if you have those mutations present, then you develop up to 7 or 10 times higher chance breast cancer or ovarian cancer. So Mira Genetics, they were kind enough that they were just mass producing these DNA kits to test women if they have the mutations in their DNA, and in the specific those two genes. That helped us a lot because first they tried to monopolize and try to make ownership of the human genome, meaning if they find the mutation, if they understand what a mutation in a specific gene does, therefore they have ownership or not. That's something that was brought all the way to Supreme Court to say no, because it happens in nature, therefore you cannot own it. However, but opened and help us to understand, okay, the technology that they apply, now we can apply it to many other genes and therefore be able to screen more people for more diseases. If you're interested in the full story, I recommend you this book. It's called Genome Defense, instead of the epic legal battle to determine who owns your DNA. Explains everything over there. But this is a classic example here, especially here in Utah, because, well, they are our neighbors. Alzheimer's disease, this is something really, really, really close to me because I've been studying this for the past almost 15 years. So Alzheimer's disease is the most common cause of neurodegeneration and cognitive decline. Some of the pathologies is the memory loss, change of mood, social withdrawal, struggling to communicate. And by now, probably all of us, regardless if we're here in Utah or in Nevada, we probably have contact with a person who have been diagnosed with Alzheimer's, or at least a type of neurodegeneration. It is really struggling because literally when you have Alzheimer's disease, your brain mass completely shrink and decays. And this happens because our neurons, astrocytes, and microglia, which are specific cells that are housed in our brain, they just get full with amyloid plaques, which is a protein that accumulates in our brain and it no longer can be withdrawn from it. And our microglia and astrocytes, which are the cells responsible to clean those peptides, they just go into a stage of they will no longer work, into microgliosis and astrogliosis. Going from a normal cell, sorry, my mouse is not working anymore. There you go. From a stage of normal cells to a non-working cells for microglia and astrocytes. In science, we have been studying the Alzheimer's disease for many, many years. And this is a complex disease. It's not like sickle cell anemia or breast cancer that one mutation will cause the gene. Here I'm showing you how through all the genes, through all the research that we have done in the human genome, we have identified multiple mutations that might be associated with Alzheimer's disease. Here on the y-axis, you can see p-values. The higher the p-value, all the way from 0 to 20, that means the more significant it is for a risk to develop Alzheimer's disease. In the x coordinates, we have our chromosomes, like all the way from 1 to 22. And we call this a Manhattan plot because, I don't know, looks like Manhattan, right? Like with all these peaks. But what is important for here to know is that all these are specific point mutations, and then this line at 5 is our statistically significant threshold. This 7.2 is our biological statistical significance. Everything above this red line over here are genes and point mutations that play a risk to develop this complex disease, Alzheimer's disease. As I mentioned to you, and there are many, as you can see, there are multiple. And now the research is moving from thinking, we're going to fix one mutation, one gene, and see if we cure the disease. We're trying to understand that we need to play in a much broader field. And my field is the immune system. And here are just pointing all the mutations or the genes that are involved in the immune system, in the cell, in the brain. Because, as I said, Alzheimer's is really complex. There are multiple players, there are multiple proteins, there are multiple cells playing a role of developing or not developing Alzheimer's disease. And at least for my backyard, where I like to play, I focus on cytokines and chemokines, which are part of our immune system because they They have a direct role with microglia, astrocytes, the peptides they form in the brain, and also neurodegenerations in the neurons. So that's what I focus my research most of the time. Talking about our DNA research, CRISPR-Cas9 is a technology that allows us to edit our DNA. It's not being used in humans as we know so far, because it can be really helpful and it can be really harmful. For example, how this works is that we have this protein Cas9 that comes and reads the entire DNA. And with this guide RNA, which comes and identifying a specific point of our DNA, and when identify that specific point, it just Cas9 come and makes a double helix or a single helix cut, and that's what it does. So, but we have engineered it in a way that after Cas9 comes and makes the cut, we can possibly come and say, okay, now I have the desired DNA sequence or template that I want you to fix. Take out that piece of DNA that is bad, and I can just put a new one, and hopefully that is, to the scientist, the right one, right? So if you think about it, sickle cell anemia, this could be a good treatment to have. But obviously Cas9 is not perfect. CRISPR-Cas9 is not perfect. It's still in development, and it's not as easy as it sounds, because then you need to have this specific treatment for every single cell that you have. And that's where the caveat comes, right? Other research that we do is epigenetics. DNA, as I said to you at the beginning, is not as simple as it is, right? There are more components that comes into place, and it is more like instead of a straight line, it's more like a 3D structure, where in order for transcription and translation to happen, many other components and proteins needs to come and being able to bend it together to be able to open the double helix, to have the right sequence to start transcribing everything. So people are trying to understand how these components, or these 3D structures, they help to modulate the transcriptions of a good or a bad copy. And if we can just being able to help to modulate the disease. Now we have the potential to go all the way to single cell proteomics, and this is something that I've been working lately. In this case, I'm just showing you here that we have checked the DNA, the protein expression of roughly 80,000 cells from the brain, and we know that, and we are able to compare it at different stages. The stages that I'm comparing right now are like metabolic stages of fed, fasted, and refed. And we can see the proteins expressed differently. Blue is they do not express, and red are really heavily active expressing. In this case, different proteins and different cell types, they behave different depending of the state that they are in. And we can just go and do the whole signature for every single cell that we have. And then hopefully this will give us information enough to being able to say, okay, how does our cells are actually behaving when we have a specific type of disease, or when we don't have it, or when we are going into a specific treatment for a disease? Are they behaving like a normal cells, or are they behaving like a sick cell, and all of that? So this is something that a lot of the science and a lot of the research is going into this. Lastly, we have some animal models organisms where, I mean, yes, we can do everything in humans, but to be honest, we can learn from the DNA composition from mouse, rats, primates, pigs, flies, yeast, worms, any other organisms, because we share some commonalities. And doesn't translate 100% to humans, but at least it's allow us to have an idea how those treatments might behave, or might will do in different organisms before we test it into humans. And as every drug or medicine that we take, they need to go by the FDA. They need to pass some regimen. And some of those regimens, they need to know, like, okay, how this specific drug behaves in any other animal organism before we put it into humans. How DNA research is shaping the future? Well, gene therapy, precision medicine is something really that is a hot topic right now, right? We understand that not every treatment works for everybody. Ibuprofen is not the cure to every single symptom that we have. Or for the people who are not from here, paracetamol doesn't take away every single disease, right? So what we're doing right now is now that we have the potential to understand how each person proteomics or proteins are being expressed or DNA mutations and all of that, we're trying to understand, okay, what about if we're trying to understand all of that to personalize the medicine that you're going to be taking. And it might not be just for a single person, but it might be that treatment for a specific group of people who behaves biologically the same. So that's the whole idea of precision medicine. And a controversial topic, I don't know where you ended up on the whole vaccination realm, but at least understanding how DNA works also help us to develop the RNA vaccines that help us with COVID and understand how how safe those are, because now we understand that RNA only lands on the cytoplasm, will never go all the way to the nucleus and change our DNA molecules. First of all, it doesn't work that way. Also, now we know that if there is a specific receptor that a virus is attaching to our body or our cells, then now we know that with an RNA vaccine, we can just create kind of like a blockage to that receptor, so that virus doesn't come in place or they cannot penetrate into the host, kind of like what the mutation for sickle cell anemia does as a natural resources, right? So DNA, I don't know, it's on our understanding of DNA and how we can use it and utilize it to treat different diseases is changing a lot and is helping us a lot to, well, save lives, to create better treatments, and to gain more knowledge of our lives. We continue discovering more and more things about ourselves, and that's fascinating because it only shows like how little we know about our own biology, right? With that, I just want to say thank you to the people, the amazing group that I work with at a university at the U, with BYU, and I want to say special thanks to Dr. Freitas because she's not only a colleague, but she's an amazing friend of mine. And with that, thank you so much for your time and attention.
Wow, thank you. That was amazing. Well, if we'd like to take some questions, if that's okay. We want to
Yes,
start in South Jordan, see if anyone there
I
has.
have. Yeah, we have one
We
here.
have one, so make sure for the recording.
Thank you. Well done, Josue. Appreciate your presentation today. Thank you. Two questions for you regarding Alzheimer's specifically. What percentage of Alzheimer's do you believe is genetic, and what is epigenetic? Number one that's my first question. And then the second question is, if you are able to identify within my DNA certain genes that make me more prone to getting Alzheimer's, what kind of therapies are available to me? If you look at BRCA, there are therapies associated. Once a woman identifies that they're at risk, they can go through certain therapies. CRISPR, as you said, is not maybe an option right now, something similar in the future, but what types of therapies are available?
Dr. Gonzalez, can you repeat the question? We are having trouble hearing.
Yes, the first question is, what
microphone.
percentage of, for Alzheimer's is related to Alzheimer's disease, both questions. The first question is, what percentage for Alzheimer's disease, what is the percentage that plays in role in genetics and epigenetics? So the genetic, we believe that it is something between, we used to believe that it was something between 20 to 40%. That would be your risk for genetics. Now we tend to believe that it's much higher, somewhere between 60 to 80, sometimes even 90% that is fully genetic related to that, at least for late onset Alzheimer's disease, meaning that's when you are diagnosed at the age of 65 or higher, and you're literally cognitive decline really rapidly after that. That's for the one that at least I study. For early onset is more environmental. It plays a little bit on the genetics as well, but it's more the environment. It's more like what we were just been talking about, like how like the nutrition that you have, the access to education that you have, in all of that, the type of physical activities that you have. Are you the person who is he the kid who just go home and go straight into the screens, or is he a little bit active, or plays some sports, or reads, or plays music, or do we have any mental exercise, right? So that, and question number 2 was what kind of therapies do we have for Alzheimer's disease now that we understand that we have some mutation risk. First of all of us, we have the same genes. Like that's like the whole concept of saying that I have a gene that will give me X or Y disease. Well, all of us, we have the same gene. What is different is the mutation, the DNA changes inside of those genes. So if we have a mutation or a DNA change that give me a higher risk to develop Alzheimer's disease, and this is specifically for Alzheimer's disease, I am sad to tell you that there is nothing available right now. We have been developing some drugs. Well, you read it all over the paper since 2000 that we've been coming up with antibody type of treatment for Alzheimer's disease, where they come and they goes into our brain and takes away that peptide. Sadly, for late onset Alzheimer's disease, when you qualify for that specific drug, you're 65, 70, and you're diagnosed with a disease, and now you can get access to that specific drug through your insurance. Otherwise, it's about $7,000 out of your own pocket. But the problem doesn't land when you're diagnosed with Alzheimer's disease. The problem is happening when you're in your 30, 35, when all those cells I showed you, like the microglia and the astrocytes, they suddenly collapse because they are not working anymore to clear that peptide that has been accumulating. And with years, then your brain loses its mass, loses its neuron connectivity, and then boom, when you're 65, all of a sudden, it's like day and night. Like you're not capable to be the same person. So we are working in the whole field, physicians, industry, academia, is trying to work, how can we do a better diagnosis much earlier? Because that's when matters, when we want to prevent it. In my case, when microglia and astrocyte, those two cells are specifically with immune response, and because there is something happening in your brain. So I trying to understand, like, what are those markers in those cells when they are going into produce into microgliosis or astrogliosis, which it will derail those cells from working properly working. How early can we catch them? What can we do? And there are several things, like CSF from your cerebrospinal fluid. You can take some measurements from those peptides that have been accumulating out, and then, but that is really invasive, right? There are cohorts in the Netherlands that there are people who are voluntarily, every 3 months, they go and they get a CSF extraction, and protein is being measured on that sense. just for us to understand when is the tipping point where like something is happening, it's not a point of return anymore. Besides that, there are a lot of literature that will suggest to you, like, I don't know, have the Mediterranean diet. Yes, it helps, but it's not a treatment. Have more active life. Yes, it helps, but it also at 65, some people also, they just do it. Also here in Utah, we have this in the Cache County, Logan area, we have this cohort of people that they have been isolated for such a long time, just because how Utah works, and then sort of have inbreeding within themselves. They've been so isolated and into people have been like, I don't know, they marry my third cousin or something like that for so many years. And we found in this population kind of like super controls. They have every single mutation that we can possibly predict that will cause Alzheimer's disease. And those people, for some reason, they don't develop it. It's something amazing that we're also trying to understand, like, okay, what else is happening? Because obviously what we currently know is not 100% true. Like, you can have all the markers to develop Alzheimer's. Like, people here in Utah, they prove us it's wrong. I have them all, and they live to their 90s, almost 100, and nothing happened. So, yeah.
Were there any other questions in Utah?
No more questions from us, so we'll turn it to Vegas.
Oh, great. Quick question, if you could just sort of add on to Dr. Moffat's question about if you're able to talk about the GLP-1 drugs and the findings in terms of their being neuroprotective and helping people either that currently have Alzheimer's, prevent Alzheimer's. I know that that's all very new. Are you able to talk about that a little bit?
Yes, I can. Do you have any specific question?
We talked yesterday about the $1 million settlements in March trial where it's Miller March. But this is what you really should know. When the other lawyers and insurance people differ, they call me. There's a reason. All that. Or lawyer. Speak.
Sorry, somebody was unmuted, so I'm sorry. Did you hear the question?
Sorry, we did not.
Yeah, we
Dr.
did not hear the
Gonzalez.
question.
Okay, the question was about the GLP drugs and the recent information that they are neuroprotective and help people that have Alzheimer's or prevent Alzheimer's. Could you talk a little bit about the GLP-1 drugs? and
Yes.
Alzheimer's? Thank you.
Yes. So just as a general concept, this is another drug that it's an antibody-based drug, and the whole function of that is that it will come and it clear the peptide out of your brain. The peptide that clears is called amyloid beta, and amyloid beta is one of the main markers for Alzheimer's disease. And producing this peptide is
You got
helping.
it?
What?
Go ahead. Somebody
Um,
again. Thank you. Go ahead, Dr. Gonzalez.
Having this antibody in your brain through this drug, it comes and it helps you to give a rest to these microglia and astrocytes. So yes, in terms, it's a neuroprotective because it allows is doing the function of these cells of clearing this peptide, and that's the reason why the company claim it is a neuroprotective one against neurodegeneration, because yes, it will last. It will improve a little bit the lifespan of a healthy person, the quality of life. That's what I'm going with. Of this patient, they will potentially develop Alzheimer's disease. It's just because the accumulation of those peptides doesn't happen as quick as it would be happening without this drug.
Thank you. We have a question on Zoom, if that's okay. Mia asks, what is the percentage of the BRCA1 or 2 mutation that causes cancer?
The percentage. I was reading this morning, as I was preparing for this talk, a review paper that came up in Nature in this year, 2024. They were saying that each mutation, each gene, they have found until now, right? When mirror generic happens, it was 1 or 2 mutations only. We have learned that we have a lot more, just like Alzheimer's disease. But there are about 10 to 15 mutations just in BRCA1 and 7 to 9 mutations that are highly risk to develop this disease. Now, to have these mutations, and this is something just to clarify, our body, our cells, they have a repair mechanism to avoid these mutations to happen. Like, otherwise, we'll be evolving like bacteria, for example. They change it all the time. We don't. Having been another Homo species in such a long time that I don't think that we're going to see a branch of humans into superhumans anytime soon. So for a person to have it's really rare to have more than 1 mutation in the genes. However, there are multiple mutations that have been associated with the risk, but a person tends to have 1 to 2 mutations at max at the same time. However, once you have these mutations, as I showed in my presentation, it's really high risk to develop cancer.
Thank you. Any other questions here? Nevada? Any other questions on Zoom? There's a follow-up question. I'm just going to read it. When he said 7 to 10 times, he means that certain mutations, depending which of those nucleotides AGTC is altered, increase your risk 7 to 10 times than it would have been. That was the comment back to Mia. Mia, did you have any other questions? Are you good? Okay. Well, Dr. Gonzalez, thank you so much. And thank you to all of our guests in person and online that have come tonight. We really appreciate everything. And this was really just absolutely incredible and fascinating. And we wish everyone here tonight, as you go along, that you have a very wonderful holiday season. And thank you again. We'll see you next time. Good night.